So in today's class, I'll be talking about construction of pBR322. This is an artificially created vector. Artificially created vector which is used in molecular biology cloning. Used in molecular biology cloning. So you don't really need to understand about the steps of how it is the exact intricate molecular biology of the creation of this plasmid. But what you really need is what this vector is and why it was created, what are the advantages, disadvantages, what things are present in this vector. So first thing is, what are artificial vectors? So artificial vectors are basically modified natural vectors. They are modified natural vectors. And from natural means they are occur in nature. For example, plasmids. They are also plasmids. And they are completely designed in vitro. So in test tube using molecular biology techniques, you create this particular vector. So these molecules are designed with the goal to enhance the cloning and integration capacity and ease of use. So if you have a look at this particular P vector which we are studying today, pBR322, you can see it has got two antibiotic resistance gene. One is ampicillin, the other is tetracycline. And it has got also the ORI sequence, replication ORI sequence. And you can see several restriction enzyme sites, and these are the multiple cloning sites here. These are the multiple cloning sites. This is where you can put in inserts. So inside the TET and AMP region, there are some enzyme sites you can clone. You can switch off one type of antibiotic resistance also in this particular thing. You can switch off, you break this ampicillin gene, ampicillin resistance goes off. It can only then survive in tetracycline medium. So then you have to use tetracycline in your selection medium. Okay. Now, a bit about the construction of pBR322. So in earlier times in molecular biology, naturally occurring vectors were used, such as poly1 was a naturally occurring plasmid in E. coli. You had the PSC, uh, uh, which is also a naturally cloning vector. It was used to clone gene. So they occur naturally in this journey. So although these vectors were having small size as required and a few restriction sites, but they lacked in the selectable or sporable marker system. You need a marker or antibiotic resistance that only the cells which have been transformed by this particular plasmid, they can survive. Only cells which have the antibiotic resistant plasmid can survive. Only cells which have the, so if it's the case of, if it's an ampicillin resistance and you grow it in ampicillin media, only the cells which have got the plasmid which has got, which is also known as selectable marker antibiotic, they can survive. So that the cell is transformed, you can select that these are transformed cells by seeing the resistance in antibiotic. So there were efforts made to construct a superior vector with improve cloning capacity. So pBR322 is one of the earliest used vectors. Earliest constructed vectors. So the pBR322, it is a purpose-built vector that is for a specific function, and it was the first widely used cloning vector. It has a relatively small size of 4363 base pairs. And we know that the efficiency of transformation is inversely proportional to the size of the vector. So the larger the vector, less chance that it is getting transformed into the particular bacteria. So the small size of the pBR322 made it hugely popular. In addition to being a multi-vector, it contained all the facilities that was lacked in the naturally occurring vectors or its predecessors, including large number of restriction sites and selectable markers, which are antibiotic. In this case of pBR322, it is ampicillin and tetracycline. So nomenclature, what does this pBR322 mean? P stands for plasmid. B. Bolivar. Bolivar was one of the lead researchers who developed these plasmids. R is Rodriguez. He was also one of the lead researchers who developed it. And 322. It is the plasmid vector model number, differentiating it from other plasmids developed in the same lab. Like they have pBR325, pBR327, but just that it has got some difference. Just to differentiate, this is how they made it separate. So essentially in the construction, major steps involved in the construction of pBR322, it was the ampicillin gene has been incorporated from the natural plasmid RSF2124 of E. coli. The TET R was derived from PSC101, which is also another natural plasmid. And the origin of replication ORI was derived from PMB1 plasmids. All these three were fused by a technique which, if you are interested, you can, it's in this slide, but you don't need it for your exam. You can study. It also contains a ROP gene, which encodes a restrictor of plasmid copy number. So this ROP gene, copy number means the number of plasmids present in one bacteria. If it is too high, then it becomes toxic for the cell. So they restrict it to a feasible amount. This plasmid also has more than 40 unique restriction sites for restriction enzyme. More restriction sites, more different types of inserts can be cloned in different regions. Out of these restriction sites, 11 fall within the sequence of TET-R gene and 6 fall within the sequence of ampicillin resistance gene. Total size 4361 base pairs, and it can carry inserts, the transgenic gene of interest, from 100 base pairs to 10 kb. And what are the advantages and limitations of pBR322? Advantages: it has got a small size, which is for because of this it can isolate, purify, and manipulation is very easy. It has got two selectable markers, easy selection of recombinant plasmids. It's more sure shot. If you give tetracycline and ampicillin in that media on these plasmids, you get an extra selection. You are 100% sure. And moderate number of copies can be generated inside a host cells. Limitation: chances of loss of vector mix population contamination due to high mobility of pBR322 via conjugation between bacteria. Suddenly you are transformed it copies and it goes into another bacteria. That bacteria also becomes transformed. High copy number can't be realized. You can't make high copy number over here. No sporable marker is present. making the selection process laborious. You don't have a X-gal blue-white selection. So, any questions? No questions? Then this is the end of this class. Thank you very much.